Respiratory system. The primary function of respiratory system is gas exchange. This is the exchange of oxygen and carbon dioxide between our bodies and outside air. But like many other systems in the body, it has many collateral functions. The nose plays a role in speech production and other audible sounds like laughing and crying. Smell is another function of the respiratory system. We have specialized cells in our nose that detect odors. One function that you might not consider right away is pH regulation. When carbon dioxide reacts with water in blood plasma, as we will discuss shortly, an acid is created. If we do not exhale carbon dioxide, this acid would build up in our blood, making it acidic rather than neutral. The respiratory system has multiple organs. We will begin with the nose and nasal cavity. The nose functions to warm, cleanse, and humidify the air. It also detects odors and amplify the voice. Within the nose, we can find a number of structures. The nostrils, also nares, are the openings into the nose. Inside the nostrils the flared area is called vestibule. Within the vestibule is the vibrissae. They are guard hairs and they trap particles and debris. They are called vibrissae because they vibrate or move. The internal chambers of the nose is the nasal cavity. The cavity is divided into right and left by the nasal septum. The cavity is formed by the vomer and ethmoid bones. Also within the nasal cavity is nasal conchae. The conchae are tissue folds that provide more surface area for the tissues to contact the air. This allows mucus to moisten and clean the air. Also, there are a lot of blood vessels in the tissue of the conchae which warms the incoming air. There are three different folds: superior, middle, and inferior conchae. Each nasal conchae is separated by a nasal meatus. They form a passageway for the air. There are three meatuses: superior, middle, and inferior, just following the three conchae. They increase the surface area inside the nose, so the air stays in the nose longer to humidify and warm up if necessary. The end of the nasal cavity is referred to as posterior nasal aperture. At the back of the septum and conchae is a small patch of specialized ciliated pseudostratified columnar epithelium called olfactory mucosa. It is used for sensory and detects smell. The cilia here are not mobile but function to trap odor particles. The respiratory mucosa covers rest of the cavity and into the lungs. It is non-sensory and made of pseudostratified columnar epithelium and has goblet cells and cilia. It also cleans the air. The visible portion of the nose is formed by nasal bone and cartilage. Pharynx is the muscular funnel-shaped structure that cleans air and is a passageway for both air and sometimes food. Leading from nasal and oral cavity, it extends from posterior nasal aperture to the larynx and forms the superior portion of the throat. There are three parts to pharynx: nasopharynx, oropharynx, and laryngopharynx. The nasopharynx is the posterior of the nasal cavity and makes a 90-degree turn downward, which helps trap particles. This portion of the larynx only passes air. It also contains the pharyngeal tonsil. The tonsils are lymphatic tissue and usually have antimicrobial function. The oropharynx is the next segment. It is inferior to the nasopharynx and connects to the posterior portion of the oral cavity. Both food and air pass through the oropharynx. It also contains the palatine and lingual tonsil. The laryngopharynx is next and superior to the larynx and esophagus. The laryngopharynx passes both food and air. Larynx is also part of throat and found in the superior portion of the neck. It is made up of cartilages that functions to keep food out of the trachea and also produce sound. The larynx is made up of 9 cartilages. The predominant one is called the thyroid cartilage and is shaped like a shield. What we refer to as Adam's apple is the peak of the ridge shaped on the anterior surface. Males usually have a large Adam's apple than females, and this is due to testosterone. The levels of testosterone in males stimulate the peak of the thyroid cartilage to grow and become more prominent. The thyroid cartilage forms a ring with the cricoid cartilage below it. It is commonly referred to as our voice box. There are few other structures within the larynx. The glottis is the opening and has vocal cords. The epiglottis is at the superior opening to larynx. It is a cartilage that closes over the glottis so that food does not get into trachea when swallowing. But adults can swallow fine without it. And sometimes food does go into the top part of trachea and then we cough vigorously to get it out. In infants, the larynx and epiglottis are higher in throat. The epiglottis help to direct milk down the esophagus so that they can nurse and breathe at the same time. Vocal cords. The vocal cords are inferior fold of the wall of the larynx. They produce sound when air flows over them. The cords contain vocal ligaments and are covered with stratified squamous epithelium. Sound production is when the vocal ligaments vibrate from air passing over them. Muscles control the vocal cords by pulling on the cartilages. Depending on their direction of rotation, varying frequencies are produced. In males, cords are longer and thicker, so they vibrate more slowly than females, therefore producing lower frequencies. Loudness is caused by force of air. Speech is formed by the shape of the oral cavity and placement of the lips and tongues. Also associated with the vocal cords are the vestibular folds. The vestibular folds do not actually contribute to the production of sound, but close off the larynx when we are swallowing. The trachea is a rigid tube supported by C-shaped rings of cartilage and commonly referred to as your windpipe. The C-shaped cartilage rings help provide support to the trachea as a passageway for air to the lung. The ring shape also allows for the trachea to be flexible as food passes along the esophagus, which runs posteriorly or just behind the trachea. The trachea is lined with the pseudostratified columnar epithelium. Recall from the lecture on tissue that pseudostratified columnar epithelium is associated with cilia to sweep things or particles along and goblet cells that produce mucus. The mucus traps air particles to further clean the air. The trachea is a physical connection between the larynx and the bronchial tree, which comes next. Bronchial tree. The next set of structures is referred to as bronchial tree. It is a network of highly branched air tubes that starts when trachea split into right and left main bronchi. The main bronchi go to each lung, one in each lung. The tube splits again so that each lung receives an air tube. This branch is referred to as lobar bronchi. The right lung has 3 lobar bronchi on each for the superior, middle, and inferior lobes. The left lung has 2 lobar bronchi, one to the superior and one to the inferior lobe. Once in the lobe, the tube will branch a number of times. These branches of the lobar bronchi and are called segmental bronchi. The main bronchi continue the C-shaped rings, but the lobar and segmental bronchi transition to cartilage plates that overlap each other. Bronchioles are continuation of the airway, but they lose the supportive cartilage. The bronchioles are transition from skeletal muscle to smooth and pseudostratified epithelium to ciliated cuboidal. The terminal bronchioles are the last branch of air ducts that act solely as an air passageway. They do not have goblet cells but still retain the cilia. Each terminal bronchiole branches into 2 respiratory bronchioles. This branch is considered the beginning of respiratory division because the corresponding alveoli are the site of respiration. In the respiratory bronchi, we start to lose the smooth muscle and cilia, and the cells start to become smaller. The next structure to follow the respiratory bronchioles lead to the alveolar ducts. The alveolar ducts are thin-walled passages. They are not ciliated and become simple squamous epithelium and end at the alveolar sacs. This is lower respiratory tract, our bronchial tree, the C-shaped cartilage rings, and the mucus escalator.
So all those pseudostratified columnar pushing the mucus up and out of the respiratory system.
Next is lungs. The lungs are divided into various lobes. The right lung has 3 lobes, the superior, middle, and inferior lobes, and the left lung has 2 lobes, the superior and inferior lobes. Since the heart lies towards the left side of the thoracic cavity, the left side has less space and therefore fewer lobes. In this diagram, we can see airway as it starts at the larynx, continues down the trachea, and splits into right and left main bronchi. We can also see the different lobes of each lung, and on the right we can see the lobar bronchi that deliver air into each individual lobe. Once in the lobe, the airway will then branch into the segmental branch. They are not labeled here. Alveoli. The alveoli is where the gas exchange will occur within the lungs. A group of alveoli is referred to as alveolar sacs. The alveoli consist of following cell types. Type 1 cells are simple squamous cells and make up the 95% of surface area. They are needed for rapid gas exchange. Type 2 cells are cuboidal cells, account for 5% of the surface area but are actually more numerous. Their job is to repair alveolar epithelium when type 1 cells are damaged. They also secrete pulmonary surfactant, which is lipids and proteins that coat the alveoli. Surfactant prevents the wall of the alveoli from sticking together, so inflating them next time will be easier. In the picture above, you can see the alveoli and alveolar sacs and the capillaries that surround them. Also notice the bronchiole, terminal bronchiole, and respiratory bronchiole mentioned in previous slides. Pleurae. Lungs also have a protective covering. They have a visceral and parietal pleura, which should sound somewhat familiar after our heart discussion. The visceral pleura is a serous membrane that covers the surface of the lung. The parietal pleura is formed by the visceral pleura folding back onto itself, opposite of the pericardium, and covers the inner surface of the rib cage and the superior portion of the diaphragm and adheres to the mediastinum, that is the concave portion of lung. The pleural cavity is the space between the pleurae. It also contains a pleural fluid that functions to reduce friction, creation of a pressure gradient, and compartmentalize the thoracic cavity. This is to prevent infection from spreading from one organ to another, while the term cavity implies this massive space. It is actually very minimal. Site of gas exchange. The respiratory membrane is the structural barrier between air and blood. It consists of the alveolar wall, endothelium of the capillary wall, and a shared basement membrane. As previously mentioned, the alveolar wall has two types of cells, type 1 and type 2 cells. We can also find macrophages, also known as dust cells. These are the most numerous cells in lungs. They wander the internal space of alveoli and connective tissue and keeps the alveoli clear and phagocytize particles like dust and bacteria. They also help if there is bleeding in the lung. They phagocytize bacteria and blood cells. These cells are then pushed up the trachea by the mucus escalator and swallowed by the esophagus. The second part of the respiratory membrane is the capillary wall. The capillary wall is made up of squamous epithelium. The entire alveoli is surrounded by a network of capillaries. The respiratory membrane is where gas exchange occurs. Respiration. Respiration refers to three different processes that occur. First is the exchange of gases that occurs between atmosphere and alveoli. This is called pulmonary ventilation. Secondly, it is the exchange of gases between the alveoli and the blood. And lastly, the gas transport, that is how gases are transported in the blood. It is the exchange of gases between the blood and body cells. Finally, the use of oxygen in cellular metabolism, as in the production of ATP, can also be referred as respiration. We are going to focus on the first two types in our discussion of respiration. Exhalation. Expiration, in contrast, is a passive process. Muscles are returning to their original position. With the decrease in space, there is an increase in pressure. The air flows along the pressure gradient and out of the body. The decreasing size of the thoracic cavity also physically pushes the air out, like when you push on a bag. There is a difference between quiet and forced respiration. Quiet is when a person is at rest, and forced is a deeper, more rapid breathing from exercise, like singing, playing instruments, etc. Sites of respiration. Respiration is based on the movement of gases down their pressure gradients. Gases always flow from higher pressure to lower pressure. There are two types of pressure found in the lungs. Alveolar or intrapulmonary pressure is pressure within the alveoli. As space in the cavity increases, the pressure here drops. Negative pressure in comparison to atmospheric pressure. Or there is less pressure here than the outside of the body. When we start our respiration cycle by expanding the thoracic cavity, the amount of pressure drops about 3 millimeter mercury. Intrapleural pressure is the pressure within the two pleuric layers. Pressure drops during inspiration as the parietal pleura is pulled away and creates a space. This is a negative drop in pressure again. It drops from 4 to 6 millimeter mercury. This will lead to the exchange of air. Air is composed of 78.6% nitrogen, 20.9% oxygen, 0.04% carbon dioxide. Few other gases make up a small amount of percentage. The atmospheric pressure is the total pressure of all the gases combined. According to Dalton's law, each gas in a mixture of gases exerts its own pressure as if no other gases were present. Partial pressure is the amount of pressure exerted by each individual gas. Partial pressure is in particular what drives exchange. As I mentioned, it works like a concentration gradient. Gases will flow from high pressure to low pressure. Let us review the respiratory cycle. First, during inspiration, the thoracic cavity is expanded by the elevation of the ribs and flattening of diaphragm. This decreases the air pressure inside the lungs, also known as intrapulmonary or alveolar pressure. In comparison to the air pressure outside the body, this is lower. The pressure within the pleura cavity also decreases, contributing the overall effect. The decrease in pressure allows air to flow into the lungs from atmosphere. During expiration, the diaphragm rises and the rib cage settles back into its position. This physically forces the air out, but it also decreases the space within the thoracic cavity. The decrease in space increases the pressure, causing the air to flow out. Let us return to the diagram of the alveoli. In the alveoli of the lungs, the pressure of oxygen is high, but the capillaries have a low oxygen pressure. Therefore, oxygen will flow from lungs to the capillaries. It is the reverse for carbon dioxide. The partial pressure of carbon dioxide is low in the lungs but high in the capillaries. So it travels from high to low along its pressure gradient. As the blood leaves the lungs, it is backed full of fresh oxygen. It is then travels to the rest of the body. The cells of the body are low on oxygen. So again, it flows from the high pressure levels in the blood to the low pressure levels within the cell. Carbon dioxide will do the opposite. Cells produce great abundance of carbon dioxide as a waste product. The cells disposes of it by having it diffuse into the blood where there are low pressure levels of carbon dioxide. Blood then returns to the heart and lungs, and the process will start all over again. The video linked in the slide demonstrates gas exchange via pressure gradients. Now we come to the transportation of gases. As was mentioned in our blood unit, oxygen is primarily transported via hemoglobin, about 98.5% of it. Recall that oxygen travels in molecular form O2, and hemoglobin can carry 4 molecules or a total of 8 oxygen atoms. Hemoglobin is called oxyhemoglobin when there is one or more oxygen molecules bonded to the hemoglobin. The remainder of 1.5% oxygen is transported as dissolved in gas in blood plasma. Oxygen dissolves in water very poorly. Carbon dioxide can actually be transported in 3 forms. About 90% of carbon dioxide travels through blood in the form of bicarbonate and hydrogen ions. When carbon dioxide enters the blood, it reacts with H2O to form carbonic acid, which then dissociates or break apart into bicarbonate and hydrogen ion. This reaction reverses at the lungs. Carbon dioxide is then free to diffuse out of the blood and into the alveoli. 5% of carbon dioxide binds to the amino group of plasma proteins and hemoglobin. Carbon dioxide does not compete with oxygen for a spot on hemoglobin. Remember, carbon dioxide binds to the globin chains. Although carbon dioxide does not compete, carbon monoxide does. Carbon monoxide binds to the heme group and does not let go. As a person is exposed to more and more carbon monoxide, their heme group are taken and oxygen cannot be transported in the blood, and the individual slowly suffocates. 5% of carbon dioxide also dissolves into blood as a gas, like it does in soda pop, which gives it the bubbles. This is a picture of gas exchange and transportation from your text. Neural control of breathing. A few points about how our breathing is controlled. The exact mechanism for setting the rhythm of respiration is not known. Breathing depends on repetitive stimuli of skeletal muscles from brain. Neurons in the medulla oblongata and pons control the unconscious breathing, while voluntary control is provided by motor cortex. The automatic unconscious cycle of breathing is controlled by three pairs of respiratory centers in the reticular formation of medulla oblongata and the pons. The medullary rhythmicity area is found in the medulla and has two groups. The ventral respiratory group, or VRG, sets the rhythm of breathing, how often you are inhaling. The dorsal respiratory group, DRG, also works on inhalation, in that it modifies the rate to adapt the varying conditions. The pneumotaxic centers are found in pons. It regulates the shift from inspiration to expiration in quiet breathing.